Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. My video today is about the uses of metformin in dermatological diseases. Metformin, categorized as biguanide medication, efficiently reduces blood glucose levels by reducing liver glucose production, decreasing intestinal absorption, and improving insulin sensitivity. Consequently, metformin effectively decreases both fasting and after-meal blood glucose levels. And in contrast to many modern medications, metformin is derived from a natural source utilized in herbal medicine and was not specifically designed to target a particular pathway or disease mechanism. Its safety and efficacy were established before in-depth mechanistic studies became available, making it a reliable therapy. Metformin is extensively used in the treatment of diabetes mellitus owing to its proven safety and effectiveness. And beyond its primary function as anti-diabetic medication, the numerous positive effects of metformin have broadened its application to various other medical conditions. Recent studies indicate that metformin can be applied in the treatment of conditions beyond diabetes mellitus, including cancer, obesity, liver diseases, and cardiovascular diseases. In dermatology, metformin is demonstrating promising results in the treatment of hirsutism, acne, hidradenitis suppurativa, acanthosis nigricans, psoriasis, and skin cancer. However, the exact underlying mechanism for these effects is not fully understood. Possible mechanisms by which metformin operates in various skin disorders include its suggested anti-hyperglycemic effects, anti-androgenic effects, antioxidant effects, anti-fibrotic effects, and anti-proliferative effects, and also anti-angiogenic effects and pro-autophagic effects. I will start with the anti-androgenic effects of metformin. Androgens are widely recognized hormones with effects on various organs, including the skin. Testosterone and its more potent reduced form, 5-alpha dihydrotestosterone, have the ability to bind to androgen receptors located in sebocytes, dermal papillae cells, the outer root sheath of hair follicles, sweat glands, endothelial cells, and keratinocytes. Androgens and their corresponding receptors are believed to play a role in the development of several skin disorders. This involvement extends to conditions such as androgenetic alopecia, acne vulgaris, hidradenitis suppurativa, hirsutism, and acanthosis nigricans. The influence on these disorders is manifested through effects on the process such as hair growth, sebaceous gland proliferation, sebum production, and epithelialization. Actually, hyperandrogenism and insulin resistance are associated with each other. Hyperandrogenism has been observed to increase insulin tolerance, which can lead to type 2 diabetes and dyslipidemia. So, increased androgens trigger insulin resistance. Also, obesity contributes to insulin resistance in patients with androgen excess disorder in many ways. In patients with severe insulin resistance syndromes, regardless of the specific disease type, hyperinsulinemia is known to stimulate ovarian androgen synthesis independently of gonadotrophins. Actually, insulin resistance is characterized by a diminished response of tissues to the effects of insulin on glucose metabolism. The impact on signaling pathways and insulin responsive tissues is not uniform, with some pathways being more affected than others. Also, certain effects beyond the metabolic actions of insulin may be overexpressed in the context of insulin resistance. And we have to know that the ovaries and adrenal glands are two examples of tissues that retain sensitivity to insulin actions, and in the presence of insulin resistance, insulin may contribute to increased androgen secretion in these tissues. So, metformin plays a vital role in polycystic ovary syndrome by reducing insulin levels, leading to a decrease in luteinizing hormone and androgen levels. This normalization of hormone levels contributes to the regulation of the menstrual cycle in women with polycystic ovary syndrome. Androgens play a key role in regulating the function of sebaceous glands by binding to androgen receptors on sebocytes, and insulin-like growth factor 1 stimulates androgen receptor signaling by decreasing what's called nuclear forkhead box01 or FOXO1 transcriptional activity through the activation of the phosphoinositide 3 kinase ACT pathway. And at the end, this will lead to activation of mammalian target of rapamycin complex 1, which is called mTORC1 signaling pathway, and to increase lipogenesis by upregulating steroregulatory element binding protein. This process results in hyperseborrhea, contributing to the development of acne in affected patients. The mammalian or the mechanistic target of rapamycin mTOR signaling pathway serves as a central regulator of cellular metabolism, growth, and survival, and this mTOR responds to various signals, including those from hormones, growth factors, nutrients, energy levels, and stress, orchestrating cellular responses accordingly. Also, insulin growth factor 1 can stimulate inflammatory cytokine responses in acne by upregulating the expression of toll-like receptor 2 and 4 in sebocytes. And that's why acne tends to worsen under conditions characterized by elevated levels of insulin growth factor 1, such as high glycemic load diet, milk consumption, and in case of polycystic ovarian syndrome. On the other side, metformin activates what's called adenosine monophosphate activated protein kinase or AMPK, leading to increased expression of glucose transporter 4 and translocation of this glucose transporter 4 to the plasma membrane. This pathway will facilitate peripheral glucose utilization and enhance insulin sensitivity, and so contributes to the reduction in insulin growth factor 1 levels. So we can understand that metformin will result in reduced insulin growth factor 1 levels, which will lead to increased availability of what's called FOXO1, and at the end, this will suppress androgen receptor signaling. Also, metformin downregulates mTORC1, which is activated by insulin growth factor 1 in individuals with acne vulgaris and hidradenitis suppurativa by mechanism that is independent of the use of AMPK. Also, anti-androgenic effects of metformin have been observed in patients with polycystic ovary syndrome. Another mechanism for metformin reduces free bioactive insulin growth factor 1 levels in patients with polycystic ovary syndrome by increasing the expression of insulin-like growth factor binding protein 1. Also, metformin inhibits the toll-like receptor 4 associated nuclear factor kappa B signaling pathway. That pathway positively regulated by hyperandrogenism. Also, metformin repress testosterone-induced androgen receptor expression in cells. So now we can understand the anti-androgenic effects of metformin and its potential effectiveness in conditions linked to both hyperandrogenism and insulin resistance. This includes diseases such as polycystic ovary syndrome, hidradenitis suppurativa, acne vulgaris, hirsutism, and obesity. Now I'm going to talk about another mechanism for metformin in dermatologic diseases, which is anti-angiogenic effects. Angiogenesis, the process of forming new blood vessels from pre-existing ones, is important for physiological repair. However, excessive angiogenesis can contribute to several cutaneous diseases, including psoriasis, atopic dermatitis, rosacea, hemangiomas, melanoma, and non-melanoma skin cancer. Angiogenesis is initiated by the release of angiogenic factors, including vascular endothelial growth factors, fibroblast growth factors, angiopoietins 1 and 2, platelet-derived growth factor, and transforming growth factor beta from hypoxic tissues. The process of angiogenesis is facilitated by the degradation of the basement membrane and extracellular matrix through the action of matrix metalloproteinases, and among these matrix metalloproteinases, matrix metalloproteinase 9 plays a significant role in promoting angiogenesis by activating vascular endothelial growth factor. And since the immune cells are a significant source of angiogenic factors, angiogenesis may occur alongside chronic inflammatory skin diseases. So in conditions characterized by persistent inflammation, the involvement of immune cells can contribute to the release of angiogenic factors and thereby promoting the formation of new blood vessels. So neutrophils contribute to angiogenesis by producing vascular endothelial growth factor A and the matrix metalloproteinases. Mast cells and macrophages release both vascular endothelial growth factor A and vascular endothelial growth factor B, while T helper 17 cells produce interleukin 17, which enhances angiogenesis by upregulating vascular endothelial growth factor A. Several studies have explored the potential anti-angiogenic effects of metformin, and recent in vivo research on cancers has indicated that metformin inhibits angiogenesis by downregulating various factors, including platelet-derived growth factor B, hypoxia-inducible factor 1 alpha, induced vascular endothelial growth factor A, insulin-like growth factor binding protein 2 and 3, platelet-derived growth factor A, matrix metalloproteinase 9, endostatin, and angiogenin. These findings suggest that metformin may have therapeutic implications not only in metabolic conditions but also in diseases where angiogenesis plays a significant role, such as certain cancers. In polycystic ovary syndrome, abnormal angiogenesis as well as insulin resistance is characteristic. Metformin were found to reduce angiogenesis in polycystic ovary syndrome by raising the level of anti-angiogenic thrombospondin 1. The precise mechanisms underlying metformin's anti-angiogenic effects involve suppressing vascular endothelial growth factor expression and reducing microvessel density through mTOR complex 1 signaling pathway. Actually, dysregulation of the mTOR complex 1 pathway governing cellular growth, metabolism, and survival can contribute to uncontrolled proliferation and inflammation. This signaling pathway is activated in various inflammatory skin conditions such as atopic dermatitis, psoriasis, pemphigus, rosacea, hidradenitis suppurativa, and acne vulgaris. Also, activation of mTOR complex 1 was observed in endothelial cells of affected skin from patients with rosacea and rosacea-like mouse model induced by LL37. LL37 activates mTOR complex 1 signaling by binding to toll-like receptors 2, creating a positive feedback loop that increases the cleavage of cathelicidin to LL37. Inhibition of mTOR complex 1 signaling by rapamycin has been shown to significantly reduce LL37-induced vasculature in a mouse model. Also, studies have reported increased expression of mTOR complex 1 in both lesional and non-lesional skin of patients with hidradenitis suppurativa and acne compared to normal controls. This suggests that dysregulation of mTOR complex 1 signaling may be a common feature in these inflammatory skin conditions. Although the specific mechanism needs further investigation, considering that mTOR complex 1 promotes T helper 17 cell differentiation, it might contribute to angiogenesis in hidradenitis suppurativa and acne vulgaris through an interleukin 17 dependent pathway. And beyond the mTOR complex 1 signaling pathway, metformin may also exert its anti-angiogenic effects through alternative pathways. As an example, in a mouse model of carotid artery atherosclerotic plaques, metformin demonstrated direct binding to matrix metalloproteinase 9, leading to its degradation. Matrix metalloproteinase 9 was reduced in a metformin-treated rosacea-like mouse model, and the possible binding of metformin and matrix metalloproteinase 9 was identified. Also, metformin inhibited the inflammatory responses induced by tumor necrosis factor alpha, interleukin 1 beta, and interleukin 17A in vitro by lowering the node-like receptor family pyrene domain containing 3 inflammasome, and considering the shared characteristics of overexpressed matrix metalloproteinase 9 and activated node-like receptor P3 inflammasome in rosacea, hidradenitis suppurativa, and acne vulgaris, metformin holds promise for potentially reducing angiogenesis in these diseases. Another mechanism of action of metformin in dermatologic diseases is the antifibrotic effects. Fibrosis in skin diseases is a natural response to injury and irritation characterized by overaccumulation of connective tissue components within an organ. Excessive fibrosis in the skin can result in pathological conditions such as hypertrophic scars, keloids, chronic cutaneous graft versus host disease, and scleroderma. Excessive deposition of extracellular matrix is linked to abnormal extracellular matrix degradation and synthesis in the context of chronic inflammatory conditions observed in hypertrophic scars, keloids, and rhinophyma. The synthesis of extracellular matrix by activated fibroblasts and myofibroblasts is primarily influenced by transforming growth factor beta signaling pathway. In addition, transforming growth factor beta, oxidative stress, mechanical tension, and various pro-fibrotic factors, including connective tissue growth factor, platelet-derived growth factor, fibroblast growth factor, interleukin 4, 5, 6, 13, and 21, contribute to the production of extracellular matrix. Transforming growth factor beta 1 binds to its receptor, leading to the phosphorylation of transcription factors SMAD2 and 3, thereby inducing the expression of pro-fibrotic genes. On the other side, metformin decreases fibrosis by inhibiting transforming growth factor beta 1 production, preventing transforming growth factor beta from binding to its receptors, and blocking the phosphorylation of SMAD3. Activation of AMP activated protein kinase by metformin can prevent the phosphorylation and nuclear translocation of SMAD3, leading to reduction in SMAD3 mediated transcription. Also, AMP activated protein kinase induces the proteasomal degradation of P300, a co-activator of SMAD2 and 3, resulting in diminished acetylation and the transcription of SMAD3. The activation of AMP-activated protein kinase also decreases transforming growth factor beta-1 induced myofibroblast differentiation. Also, the antifibrotic effects of metformin are mediated by the inhibition of hypoxia-induced factor 1 alpha dependent fibroblast activation in hypoxic conditions and through downregulation of what's called P13K FOXO3 pathway. Metformin has been investigated as a potential therapeutic approach for keloids due to its antifibrotic molecular mechanisms. In studies, metformin demonstrated a reduction in the extracellular matrix components, including collagen type 1 and 3, fibronectin, and elastin within keloid spheroids. In a separate investigation, metformin was found to prevent the hypoxia-inducible factor 1 alpha dependent epithelial to mesenchymal transition of keloid fibroblasts. Additionally, attempts have been undertaken to develop a multi-layered 3D scaffold that releases metformin, aiming to reduce fibrosis and enhance wound healing. So, additional research is necessary to explore the antifibrotic effects of metformin in conditions such as acne vulgaris, hidradenitis suppurativa, and rosacea, where common pathways in fibrosis exist, and these conditions are recognized for their tendency to result in scarring. Another important mechanism of action of metformin in dermatologic diseases is the antioxidant effects. Oxidative stress resulting from the generation of reactive oxygen species is a key factor in the development of diverse skin diseases and the aging process of the skin. Various skin cells, including keratinocytes, produce reactive oxygen species in response to stimuli such as inflammatory cytokines, ultraviolet radiation, air pollution, drugs, foods, and cosmetics. Reactive oxygen species damage cellular components including DNA, lipid membranes, proteins, and other macromolecules within the cells. Also, reactive oxygen species activate signaling pathways associated with activator protein 1, mutagen activated protein kinase, and NF-kappa B and ACT. The oxidative stress in the skin is counteracted by antioxidants. Within the skin, endogenous antioxidants such as superoxide dismutase, glutathione peroxidase, glutathione S-transferase, catalase, vitamin C, vitamin A, vitamin E, sulfahydryl groups, glutathione, melatonin, carotenoids, flavonoids, coenzyme Q10, and selenium play a vital role in neutralizing the effects of oxidative stress. Multiple studies have reported diminished activities of superoxide dismutase and glutathione peroxidase, these are antioxidants, in individuals with acne when compared to control groups. And although linoleic acid has been identified as a suppressor of reactive oxygen species production, its concentration were observed to be lower in acne patients. Also, individuals with acne displayed lower levels of serum vitamin A and E. Another issue is that the levels of what's called ischemia modified albumin, which is a marker formed under oxidative stress, were found to be elevated in both acne vulgaris and hidradenitis suppurativa. Also, the increase in ischemia modified albumin was proportional to the severity of the respective diseases. Also, thiol, an antioxidant featuring sulfahydryl group, undergoes conversion to disulfide in the presence of oxidative stress. In patients with hidradenitis suppurativa, the proportion of disulfide form was observed to be higher compared to the control groups, indicating a potential elevation in the oxidative stress among these patients. In individuals with rosacea, there was an elevation in oxidative stress levels as indicated by increased levels of ferritin and the plasma melanoaldehyde, along with the reduction in antioxidant potential. LL37, a significant factor in the development of rosacea, induces the generation of reactive oxygen species by activating nicotinamide adenine dinucleotide phosphate NADPH oxidase and mobilizing intracellular calcium. Metformin demonstrates antioxidant effects by activating AMP activated protein kinase, which is a key regulator of reactive oxygen species. Under oxidative stress, AMP activated protein kinase promotes antioxidant production by activating FOXO and suppressing mTOR signaling. Also, metformin reduces reactive oxygen species production by inhibiting mitochondrial complex 1. Also, metformin has the potential to enhance its antioxidant effects through increased autophagy, enabling cells to adapt to oxidative stress. Additionally, metformin decreases melanoaldehyde levels and boosts the expression of antioxidant enzymes, including glutathione S transferase and superoxide dismutase. So, as a conclusion, metformin, a widely utilized hypoglycemic drug known for its established safety, offers various advantages beyond glucose reduction. And given its anti-androgenic and anti-angiogenic, anti-fibrotic, and antioxidant properties, metformin represents a promising therapeutic option for conditions such as acne vulgaris, hidradenitis suppurativa, and rosacea. Here is the main reference that I have used in this video. It's published in Acta Dermaimmunology in December 2023, and its title is Metformin, a potential treatment for acne, hidradenitis suppurativa, and rosacea. Another reference that was published in World Journal of Diabetes 2021, and its title is Role of Insulin and Insulin Resistance in Androgen Excess Disorders. Another one is Metformin, a review of potential mechanism and therapeutic utility beyond diabetes, that was published in 2023. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.